Which camera? That one, right? Okay. It gives me tremendous pleasure to welcome you here to Nat Young's her master's thesis. Nat is one of our pandemic babies. So she joined the lab at the worst time that you can imagine, actually, which January 2020. So she was in the lab for a little over 2 months before things started to break down. And in spite of that and the other traumas of 2020 and since, here we are. And she's transformed that time into a lovely study that you'll all hear about today. I wanted just to say one little personal note. There are a number of reasons why I'm especially pleased about Nat and her progress. And one of them is that when I started out with beetles, the first group I fell in love with was weevils, actually. And so that's long deep in my past, but it's also lovely to have someone who grew up about 1 hour from my hometown in Canada. So in any event, let's start with the show. Nat.
Without further ado, please hold your applause, thank you. Got it. And okay, hopefully everything's working. So welcome. Today I'm going to talk to you guys about the phylogeny and microbial diversity of Starius and related weevils. Starius, who are they? What are they? Why do we care? Starius are weevils, which are in the family Curculionidae, subfamily Molotini, and the subtribe Plincna. And in particular, these are some examples here of what Starius looks like. And I first started getting excited about Starius in general because I was working at the Natural History Museum. My mentor there was Lourdes, pictured here, and we were working on a project looking into the morphological diversity in weevils. And for those of you who don't know, there is a lot of morphological diversity in weevils. There's an astounding amount. So on the screen right now is a single genus, and those are all different species, different morphs of the same genus. So we were working on that project, and I started getting really excited about just how much there was to explore in Molotini, or in weevils, and in particular in the subfamily Molotini, where there's a lot that we don't know about who's related to who in that subfamily. And I'm just going to make sure I think I'm getting feedback. Oh, great. Okay. We do know that about a quarter of all known animals on Earth are beetles, and of those, the greatest proportion of beetles are weevils. So we're talking about kind of an evolutionary biologist playground in terms of trying to ask and answer questions about this group that has dominated the globe in a lot of senses. But as a master's student, where do I start with such diversity? And so when I moved up to the Pacific Northwest, I started getting excited about Starius tenoides. They are weevils that live under driftwood on ocean shores. So if you're in Oregon or Washington on the coast and you flip a log, you might be able to find these little guys. And so before I get too full-blown into the talk, I do want to just give a brief outline of what we're going to be doing today, because I ended up kind of going in lots of different directions with this project, as my committee members know. And what came from that is that I asked questions both about the phylogeny of Starius and related weevils, and I was also asking questions about the microbial diversity. So we're kind of here, we're talking about the introduction, and I'm going to start launching into the weevil phylogeny, and there will be an independent backgrounds, methods, and results for the weevil phylogeny. Same thing for the microbial community, and then I'll do a brief conclusion. So we are now here, weevil phylogeny. So we had Starius tenoides, that original driftwood weevil, and these are the close associates that are known to this weevil. So it's hypothesized that Starius multituberculatus and Starius quadrituberculatus are closely related, as well as two other genera, which are Gastrotaphorus and Lobosoma. So 3 genera on the screen right now. There's another group that's also closely associated or sometimes associated with Starius, and that's Stromnius. Now, there all in that subtribe that I briefly mentioned before, Plynthina, and they all live in North America. And so sometimes they get kind of lumped together, even though, as I'll talk about soon, Stromnius has a bunch of differences in comparison to the prior three genera. But because I'm going to be talking about all of them and asking questions about all of them, I will just shorten it to GLSS. So every time I say GLSS, know that I actually mean weevils that are considered Plynthina in North America. And that's just kind of a quick way of going through their genus names: Gastrotaphrus, Lobosoma, Asterias, and Stromnius. GLSS. Okay, so my first study aim that I want to talk about today. In terms of the weevil phylogeny, I wanted to determine if that group, the GLSS, is a clade. What, if any, morphological characters are unique to that group. And then I also wanted to look at molecular characters. How divergent are they in terms of DNA sequence data? And then I wanted to also look farther out and determine whether the GLSS ally with other Plynthina from around the world, given the molecular and morphological characters that I find. So this is kind of my hypothesis tree. This is the starting point. So I don't really know, beginning the project, how I think Gastrotaphrus, Lobosoma, and Asterias are going to be related to one another, but I suspect that Stromnius is going to be less closely related than the other three. So I'm suspecting that Stromnius is going to be an outgroup to Gastrotaphrus, Lobosoma, and Asterias, and then I'm expecting there's going to be some other Plynthina from around the world around that, and some other molluscine weevils, etc. And the reason for that hypothesis is that Stromnius are fairly different in terms of morphology and distribution. So distribution-wise, Asterias, most Asterias and Gastrotaphrus have this Pacific Northwestern distribution I mentioned. So it goes up into Alaska. There's some that are found in Northern California and everywhere in between. Mostly restricted to the shoreline, although they also extend into the coastal rainforest. There are two exceptions. So Asterias tenoides and Lobosoma both follow an amphi-Pacific distribution, which is just jargon, meaning that they can be found in Japan, Russia, the Aleutian Islands, Alaska, et cetera, down the Pacific Northwest, again, coastal. And for the rest of the talk, I will be focusing solely on Lobosoma hirudum, because even though Lobosoma as a genus has that distribution of amphi-Pacific, two of the species in Lobosoma are only found in the Palearctic region, so in Japan and Russia. Those are Rossens and Curlens. So for my talk, I will be focusing only on hirudum, which is the only species that's found in North America and has that kind of typical Pacific Northwestern distribution. Okay. Stromnius, in comparison, they move a little bit farther inland. They're still in Pacific Northwestern kind of coastal distribution, but not as restricted to the coastal rainforest. And one species, Stromnius shermani, is found super inland. It's found in Tennessee, North Carolina, and Western Virginia at high altitudes. As we know, the Appalachia region has super high mountains as this, my little mountain climbing weevil. In terms of morphology, there's a lot that weevil people might see at first and be like, oh, they're very different. Like Stromnius versus Gastrotaphrus, Lobosoma, and Asterias, very different. But the easiest one I could give you guys an introduction to right now is that in general, Gastrotaphrus, Asterias, and Lobosoma are smaller and Stromnius are much larger. And it's actually a little bit dramatic in terms of weevils. So Gastrotaphrus, Asterias, and Lobosoma will never be larger than, or have not been discovered to be larger than 6 millimeters, and more often than not are about 3 millimeters in length, whereas Stromnius are always larger, have only ever been found larger than 6 millimeters, and more often than not are going to be about 10 millimeters in length. So it's a bit dramatic if you are a weevil that's that small. But now I need to collect data and find out what I can find out about these guys. So I went out to the field. Again, it was the pandemic, so I was a little bit restricted in terms of where I could go. But I did most of my collecting in Oregon and Washington, and the rest of the specimens I got on loan from various museums, such as the Canadian National Insect Collection and the University of Alaska Museum. This photo is from the first, this is the view from the first time that I collected Gastrotaphrus. Gastrotaphrus was not that large. But I was out here on the beach, and this was during like the main deepest, darkest time of lockdown, and the border was closed, so I couldn't go home to Canada. And I really missed my family. And Gastrotaphorus I actually found at the very northern part of Washington on the coast, and I could see Canada in the background. So that made me very happy. This little strip of land is my home and native land. There was also some further complications with collection in that I had a hard time identifying Stereas to begin with, and I also had a hard time finding Stereas tenoides, the one that I was the most excited to find. And actually throughout the project, I never found Stereas tenoides. But luckily, David and his family came to the rescue, and so I was able to include live caught specimens of Stereas in this project because they found Stereas tenoides.
One.
Stereas tenoid D. Stereas tenoid. Okay. In addition to this, I included specimens that kind of canvassed Plynthona from around the world. And I also included specimens that were important to Stereas for taxonomic reasons or were from a similar morphology and niche. And from that, I collected molecular data. I extracted DNA, and I did so for kind of 3 categories of genes. So I did that for ribosomal nuclear genes, which is I included 18S and 28S, mitochondrial gene, CO1, and nuclear protein coding genes, arginine kinase, and EF1 alpha. And from that, I did some data bioinformatics exciting magic, where I made an alignment and did a maximum likelihood tree search and found a Weevil phylogeny. So let me show you what I found. A lot. Don't worry, I'm going to zoom in a second, but essentially what I found was this giant maximum likelihood tree. So this tree, what you're looking at here is all of those genes I just mentioned together, concatenated, and now into a tree. And the colors that we're looking at are red is for the GLSS, yellow is for other Plynthona from around the world, and blue is from Molotini and various closely related weevils to Molotini, and gray is Mayo group. And so in total, I had 80 specimens in my final phylogeny, 41 of which I had extracted DNA from, and the remainder of which I got from GenBank from online sources. So let's zoom in a little bit. And I hope the thing that is kind of most obvious from this image, let me just move this over here, is that big GLSS clade, the red group, is split in 2. So we have Gastrotaphorus lobosoma and Stereas here. Hopefully you can see my pointer way up here. And then way down here is Stremius. Now, just the distance on the tree doesn't say anything about relatedness necessarily, but what does say about relatedness is just that there are many clades of other weevils that come between Gastrotaphorus Stereas and lobosoma and Stremius, which is way over here. So there's lots of stuff going on in between here that would imply to us that they're not one singular clade. Stremius doesn't belong with Gastrotaphorus lobosoma and Stereas. I'm going to break it down a little bit more. And so I'm going to show a bunch of these during the presentation. So I just want to tell you exactly what we're looking at here. So these tables, what I'm trying to show is I'm breaking down all of the genes in terms of how much support or confidence I have for the clade in that gene. And we do that using bootstrap support, and bootstrap support is on a scale from 0 to 100. So if it's 100, it's really, really high support. I'm really confident that that clade is a well-supported clade, that that maybe relationship is valid. And I use color coding. So black is going to indicate that it's good. There's really high bootstrap support. It's 90 or above. Gray is that it's moderate, it's between 50 to 90. White is that there's okay support. It's between 0 and 50, but importantly, the clade is in the maximum likelihood tree. So bootstrap support doesn't necessarily mean that the clade is lack of bootstrap support doesn't necessarily mean that the clade isn't in the maximum likelihood tree. So white is still fine. Pink is weak support. So there's maximum likelihood tree has a contradictory clade. So it doesn't have that clade in the maximum likelihood tree. And the bootstrap support of a contradictory clade, not the clade that I was investigating, has moderate support of about 0 to 50. And finally, red is bad. Not only is that clade not present in maximum likelihood trees, there's not a lot of bootstrap support, but there's really high bootstrap support for a contradictory clade. And finally, the way that we're looking at it is the first value here is the bootstrap support for the clade, followed by a comma, and the second value is bootstrap support for a contradictory clade. So, long story, what we see here is not only in all genes was there not that clade in maximum likelihood tree, but also it doesn't happen in any of the genes individually, and there's weak support at best, and really there's like low moderate support for a contradictory clade. And if we look at Gastrotaphrus, lobosoma, and Asterias without Stromnius, we find a little bit more support. So in 2 of the genes, EF1 alpha and 28S, there's some moderate support. And then in 1 gene, 18S, well, actually kind of 3 genes, so 18S, EF1 alpha, and 28S, the clade is found in maximum likelihood trees. In arginine kinase and CO1, there's not very high support for the clade, but there's also not high support for a contradictory clade. And importantly, I want to note that for arginine kinase, I only had 1 specimen of lobosoma that I was able to sequence data for. So it kind of makes arginine kinase a little bit of weaker information in terms of relatedness of like how lobosoma would fit into this equation. Another thing that kind of came up from the tree that was interesting is that Asterias is not monophyletic. So here's where Asterias is, Asterias multituberculatus, quadrituberculatus, and tenoides. I would have expected this to be just 1 clade, but instead in all instances, Gastrotaphrus barberi interrupts that clade. Let's take a look. And so we see that in all genes, not only is Asterias not a clade, but also there's really high support for a contradictory clade. And that contradictory clade includes Gastrotaphrus, and we're pretty confident about that. So high bootstrap support in 2 or at least 1 gene, and then supportive in all genes. It's in the maximum likelihood trees for 18S and CO1, and moderately supported in 28S. Okay, cool. We found some things. Now, next part of that question is, does the morphology concur with the inverted by blushing? So I wanted to know, are there any morphological traits that would kind of combine Gastrotaphrus, lobosoma, and Asterias and show that they are different from Stromnius? Is there any unique derived characters that they have? And, you know, the kind of long answer short is that no, there's no characters that are phylogenetically informative that unite Gastrotaphrus, lobosoma, and Asterias. And so what I mean by that is that there are characters that differentiate Gastrotaphrus, lobosoma, and Asterias from Stromnius, right? We talked about size, there's other features, but a lot of those features are convergent examples of convergent morphology and are associated with flightlessness in weevils, which occurs a lot in weevils. So we have traits like reduction of metadepisternum and scutellum and all these fun words about weevils that I'm sure you guys care about a lot, all of which are associated with flightlessness. And not any of them are really just in these guys make them unique and different from Stromnius. There was 1 trait of interest, which is this projection of the elytra that was a derived character trait, or I believe it's a derived character trait for weevils, at least in this area, so Molotini plynthina. This trait isn't seen a lot. It looks to be a derived trait. I'll zoom in a little bit so you can see what I'm talking about. There's just this little tooth right here. It was only in lobosoma and not Gastrotaphrus and Asterias. And so it doesn't tell us much about like Stromnius versus Gastrotaphrus, Asterias, and lobosoma. But what it does tell us is that there's something different about lobosoma and Gastrotaphrus and Asterias. And to me, that starts to suggest that Gastrotaphrus and Asterias, there was that clade where they were always together. Now there's some morphological evidence to suggest that maybe they're actually the same thing. Maybe it's just one Asterias clade. Gastrotaphrus might be another species of that clade. But I couldn't determine if just based on that information, whether or not Stromnius was certainly different based on morphological characters. So, okay, well, maybe I can help to understand it by looking at, are Gastrotaphrus, lobosoma, and Asterias more closely related to something else? So I took a look at Micromastus gracilis. Another driftwood weevil is in California, another Molotini. Weak support across the board, no morphological characters that are shared derived traits. not likely micromastis as sister. Darumazo is a Japanese molotini that's been sometimes hypothesized to be closely related to these guys. Took a look at the molecular data, not strong support, but also I was missing a lot of data. I didn't have 18S arginine kinase and EF1 alpha, so weak support, but who's to really say? But in terms of morphological evidence, I also still couldn't find any shared derived characters. No. Finally, Limerobius and Hydrampus, which might seem like to the Weevil folks in the room, okay, this might seem like an interesting choice as something that I would start to investigate because these guys are really large Mollitina from New Zealand. They wouldn't necessarily be your first pick as a sister group to Gastrotraphus lobosoma and Asterias. But as you can see, it came out in the maximum likelihood tree for all genes together that they were sister. And I was like, what the heck? But looking at the molecular data, there was only one gene for which I had data for Gastrotraphus lobosoma Asterias and Limerobius and Hydrampus. There was weak support in that in that gene. And then the fact that there's moderate support, like, it's fine, okay support in all genes, that it came out in the maximum likelihood tree for all genes. To me, this now more suggests, because there's also lacking data in every other gene, it more suggests that there's a lot of uncertainty in terms of where Gastrotraphus lobosoma and Asterias should be placed, not necessarily that it's certainly going to be Limerobius and Hydrampus, because there's not a lot of support in the only gene that there is there. It's kind of just like, well, out of the options you've given me to make a clade, I guess with Limerobius and Hydrampus, not that it's actually something that's super strongly supported. So I wouldn't say there's a lot of evidence there for Limerobius and Hydrampus. There's also a lot of evidence to suggest that Limerobius and Hydrampus have other close relatives that are not Gastrotraphus lobosoma and Asterias. Okay, so couldn't figure that one out. Maybe Stremnius is more closely related to something else. So let's take a look. Well, in the maximum likelihood tree, Stremnius was more close to something else. It was close to some other Plinthina from around the world. So things like Plinthus and Hoploptridius. There's also this guy, Eudosominas, in there. That clade, again, doesn't have a super high amount of support. There's moderate support there's kind of fine support with all genes, but it doesn't really correspond to the individual genes. So 18S and CO1 kind of have weak support at best. There's not high support for a contradictory clade, so I guess that's something, but I also didn't have information for a lot of the genes there either, so kind of not enough information to say. But looking at morphological characters, there is one shared derived trait that Plinthus, Hoploptridius, and Stremnius do seem to all share. And it's just kind of this character of the eye here. And so maybe why is suggestive? I don't know. And that's where we're at with Weevil phylogeny. That's the things that I found out with Weevil phylogeny. So what else can I find out about these guys? And so here I started to look into the microbial community. Here we are in the microbial community section. So other things that we know about these weevils in general is that they are small, they're flightless, and they eat dead and dying wood. And the dead and dying wood part is important. I'm going to start using this word a lot, saproxylic or saproxylate, and it just means an organism that eats dead and dying wood. And it's significant for weevils. It's significant for a lot of things because wood is not easy to eat. Lignocelluloses make it difficult to break down wood. Diterpenes, which can be found in sap, can be a deterrent for insects to eat wood and can also cause death of the weevil if they get caught in it. And it's just a defensive compound. And so weevils can utilize symbiosis with bacteria to help break down some of those difficult to break down components of the wood. And what is known is that weevils in nutrient-poor habitats tend to have higher richness of bacterial taxa and also higher relative abundance of those taxa. And so what I mean by that is if you're a weevil, you're eating a nutrient-rich resource, you will probably still have endosymbiotic bacteria, but you might not have lots of different types of endosymbiotic bacteria trying to break down your already rich nutrient food source. Whereas if you're eating something nutrient poor like wood, you might have a lot of varying different types of microbes to help you to break down that material. And so when I was thinking about it for this project, I started thinking about it in terms of stereostenoides. There's evidence that the driftwood is going to be a nutrient poor resource. And so I'm kind of expecting that the stereostenoides, the beach restricted ones, are going to have a higher abundance and more diversity of microbial taxa, and that the remaining GLSS that aren't as restricted to the beaches, that kind of have a little bit more into the coastal rainforest, might have a more conserved microbial community. So I guess I should also point out that these little jelly beans here, I'm kind of implying that these are the bacteria, these are the microbes. So here's, stereostenoide with all its different microbes. It's got a huge, nice, vast, varied microbial community, and the rest of the GLS have microbes associated with them, but maybe they're all the same or similar microbes. Great. Other things we know about weevil microbes, microbiology, is that there are 3 primary endosymbionts that are known to weevils that are Nardinella, Crepulionophilus, and Sodalis. Let me tell you about them. Okay, so Nardinella. Things we know about Nardinella is that they have a super reduced genome, and the functions of Nardinella, the endosymbiotic strain, well, there's only endosymbiotic strains, the function of Nardinella is really reduced such that the only biological product that they can really provide for the weevil to help them is tyrosine. And tyrosine is important to weevils. Without tyrosine, weevils can't, there's cost to being able to, actually, let me rephrase that. So tyrosine helps weevils to harden the cuticle and the exoskeleton, so like to make a really tough outer shell. And without tyrosine, there's a lack of that ability. So we're seeing like really soft exoskeleton. It can be crumpled, it can be deformed, and that has cost to their fitness overall. Also, something to note about Nardinella, and something I'm going to start talking about a lot, is a bacterium. So it just means that there's a specialized organ where Nardinella is housed. So, the weevil might be conserving, trying to hold on to that microbe. And in weevils, it's known that that bacterium is likely associated with the foregut and the midgut. And we also know that in some weevils, in that same bacterium, you're going to find Sodalis and not Nardinella. So it looks like Sodalis is replacing Nardinella in some groups. Sodalis, there is a free living strain of Sodalis, and it has a nice chunky genome. The endosymbiotic Sodalis is also really reduced in comparison to the free living strain. But it can provide a little bit more in terms of biological resources or usable resources to the weevil. So it can make amino acids, such as tyrosine, phenylalanine, vitamins, and things that can help it to break down wood products. And that can help it to strengthen its cuticle as well. Then our final primary endosymbiont is Crepulionophilus. Crepulionophilus is a little bit different in that, yes, it has been found to replace Nardinella in some weevil groups. However, the bacterium is in a different area. So the take home from that is that in some weevil groups, Sodalis is replacing Nardinella, and in some other weevil groups, Crepulionophilus is replacing Nardinella. But the evolutionary path to that replacement seems to be different. It's a different mode of takeover, essentially. And here's just a little bit closer up of looking at the bacterium of Crepulionophilus in a different area, or the bacterium of Crepulionophilus hosting organisms. Okay, let me go into some brief evolutionary context. So this is a timeline for weevils, not the microbes. What we know is that ancient weevils were likely saprophilic, so eating dead and dying wood. Way back here in the Jurassic period, things that diverged then, there was a lot of examples of saproxyle. And then there was a big explosion of weevil diversity that coincided with an explosion of the diversity of flowering plants, angiosperm dominance. And there was another explosion of weevil diversity following a mass extinction event. And so in between kind of this explosion of flowering plants, we have a lot of explosion of diversity of weevils occupying a new niche space, the flowering plant area. Then following this mass extinction event, we see some weevils returning to a saprozoic environment, eating dead and dying wood. And so it's been hypothesized that association with some microbes was what's helped to facilitate niche shifts in weevils. When we're looking at Nardinella, what we know is that the phylogeny seems to be closely correlated with the phylogeny of the weevil. So if we have closely associated or closely related weevils, and they both have Nardinella, those strains might also be closely related. So here we have 2 molotine weevils and 2 closely related strains of Nardinella. Whereas if it was a distantly related weevil, so if we had a Dryopteridine weevil and a molotine weevil, the strains of Nardinella would also be distantly related. Here's a strain 4.1 of Nardinella being distantly related to strain 4.1. In Sodalis, the relationship is a lot more promiscuous. So you might have a strain of Sodalis, 2 strains of Sodalis be closely related, but be housed in distantly related weevils. So it's just a little bit all across the board here. We get a lot of a mismatch of phylogenies, as it were. And then finally, Curculionophilus, it does seem to show evidence of cophylogeny, but it's important to note that it's only been found in Curculionini. So it's only been found in 1 group of weevils. So in that group of weevils, yeah, there seems to be, it follows that closely related strains of Curculionophilus are in closely related weevils, but we don't really know what that would look like if they were to be found in other weevils. Foreshadowing. So my study aims for this part of the project were to characterize the microbial community in Sterias, Tenoides, and other sample weevils. And I wanted to ask, is variation in the microbial community dictated more by ecological niche? So if we're thinking about the beach or the forest, or the phylogeny of the weevils. And then do the GLSS possess endosymbionts that are known from other weevils, like those primary endosymbionts I was talking about? And then I also wanted to track the occurrence of primary endosymbiontic weevils as they occur in the weevil sample. And my hypotheses. So firstly, I hypothesized that all the weevils in my sample that are saprozoic will have the key endosymbionts, either Nardinella, Sodalis, or Curculionophilus, since association with symbionts may have allowed them to colonize deadwood environments. I also hypothesized that the phylogeny of the weevils will be a greater factor in patterns of Nardinella, the one that seemed to show dense cophylogeny, but ecological niche will dictate the richness of microbial taxa. And finally, that Sterias Tenoides will have a more rich microbial community since its food source is nutrient poor, and weevils that are less restricted to the shoreline niche will have a more conserved community. Time to collect data. So I used the same collection methods for this part of the project. I used a subset of only 14 specimens of my core group of Gastrotrappus, Sterias, Lobosoma, and Stromnius. And use those for 16S amplicon sequencing. I will asterisk this by saying that I included not only freshly killed live caught samples of weevils, but I also included museum collected specimens, so things that have been housed in museums for a long time, well, a somewhat long time in cabinets housed dry. And so I did have a little bit of concern going into this that might impact the microbial community. Maybe things that were stored in a box for a long time might have different microbes that I could detect at least. I also included 3 outgroup specimens. So I had Micromastes gracilis and Emphiastes fusicola. Both of these are weevils that are also associated with the shoreline niche and in the Pacific Northwest. And then finally, Curculio, which is one of those Curculionini weevils that might have Curculionophilus for me to compare to. What I did, I took whole body insects, I extracted the DNA from them. And then I targeted the 16S region, the 16S gene, sorry. And the thing about that is that if for the 5 genes that I was using for the weevil phylogeny, I'm extracting them from one single weevil, and I know that that is the gene from that single weevil. With 16S amplicon data, that's not the case. There's lots of microbes that are going to be on that weevil. So I'm amplifying lots of snippets from lots of different organisms. And so I get all of these little 16S amplicons in return, and I have to do some bioinformatic magic to then assign all of these little snippets to a given organism. And so I'm going to say, oh, all of the little red small snippets are microbe A, yellow is microbe B, blue is microbe C, etc. And so I get all of my little clusters of 16S. And I then use a naive Bayesian classifier to try to classify what microbe do I think it is. And I was immediately having an issue, which was that I was not getting a lot of weevil endosymbionts identified, none. And also I was getting Sodalis, but only the free living strain. Remember, there was that big genome free living Sodalis. And I was like, what the heck? That's surprising to me. But then I realized that it's likely because the training set that I was using did not have weevil endosymbionts in it, which would be an issue. I also then started realizing that there were not a lot of databases out there used for microbiological studies, such as RefSeq or RedBio, also didn't have symbionts known to weevils. So I had to make a custom training set. And after I made a custom training set, I was able to identify weevil endosymbionts in some of my specimens. But this also made me a little bit concerned. This is like another caveat to my study is that, okay, well, now I've seeded in a whole bunch of weevil symbionts to this data set. And now I'm kind of worried that that was biasing the results. That, okay, I'm going to find weevil endosymbionts if I throw a whole bunch of weevil endosymbionts in there. So let me just show you what I found, and then I'll turn back to talking about some of those caveats and how I interpreted the data. So what I did find was that Asterias tenuides and Gastrotaphoris had high relative abundance of Nardinella. Asterias multituberculatus also had Nardinella, but super low relative abundance. Just 0.1% of its microbial community could be attributed to Nardinella. Asterias biotuberculatus, I couldn't find any endosymbionts known to weevils in that organism. Lobostoma, I found Sodalis. So no Nardinella and Sodalis, whereas the other three had only Nardinella, no Sodalis. In Stremnius, I found all Stremnius had Sodalis. So here's Stremnius tuberosus, Stremnius carinatus, and Stremnius germani. But interestingly, Sodalis is the blue bars, and we're looking at a chart of relative abundance. So the higher the bar, more microbe in their community. But interestingly, in Stremnius carinatus, I found not only that it had Sodalis like other Stremnius, but it also had high relative abundance of Nardinella. This was found in one other weevil group in my sample, which was Amphiastes, had a very similar distribution where they had high relative abundance of Nardinella and also had Sodalis, so they weren't like completely negatively poor, or they weren't, only presence absence of Nardinella versus Sodalis, but it was about an 8:2 type ratio in terms of amount of Nardinella to Sodalis. Finally, in Stremnius germani, I found really high relative abundance of something. But what is it? Okay, I found something, and I found something cool. I didn't know what it was. I knew that it was in high relative abundance, and I knew that the naive Bayesian classifier was calling it Herculeonophilus. Remember the foreshadowing from before. Okay, what is it? So I had to take my microbes that I found in my sample weevils, and then I also created a matrix of known symbionts to weevils. And my favorite thing, I made another phylogenetic tree. And okay, I'm just going to zoom in on the tree so you can actually see it. Hopefully you can see a little bit from the back. So here's part of the tree. And what we're looking at here is whatever the new symbiont is. So in black, we're looking at symbionts that I kind of collected from different databases that are known symbionts to weevils. In blue are the ones that I had sequenced. And so we see some strains of the blue Sodalis that were found, identified by the naive Bayesian classifier, and then this really long branch, and then a bunch of this, whatever this is. Okay, I was confused because maybe it's Sodalis, but also that branch length is suspect. So I used a different tool, the BLAST basic tool alignment search tool, which it kind of takes, it's a comparison that takes out some of our phylogenetic context. So it really just compares sequences on similarity of like the letters, exactly like what is the sequence, and not really considering other context as much. And so it makes an alignment just on the sequences, and this is really small, sorry guys, but essentially what I did find was that all of those sequences that I just showed you on that super long branch, all identified firstly as Herculeonophilus. So the highest percent identity was with Herculeonophilus. And Sodalis, not as much. And yeah. Then additionally, something else I found was that there was two, sorry, I should go back really quickly and just say that beyond them being associated with Sodalis, something that I was confused about was that, yeah, the naive Bayesian classifier classified them as Herculeonophilus. So I'm looking at this and I'm like, there's a lot of incongruencies. I do some follow-up searching. I find that it seems like they're Herculeonophilus, even though they're the Sodalis in the maximum likelihood tree. And then I also found that some other things that were found in Stromia shermani in really high relative abundance and seem to have very similar sequences to the Herculeonophilus identified symbiont aligned in the maximum likelihood tree with Herculeonophilus with some high bootstrap support. So the bootstrap value there was 100. Sorry. And so ultimately, from here on out, I decided to start calling that symbiont a provisional endosymbiont near Herculeonophilus because there was a large divergence between Sodalis and that new symbiont. There was weak bootstrap support for Sodalis in the new symbiont. There was a high similarity of the new symbiont to Herculeonophilus using blast. And also the other most highly abundant microbes in Stromia shermani were classified as Herculeonophilus. Great. What about other endosymbionts and what that looks like in the tree? So we're going to zoom in on a portion that has Nardinella. And I just want to remind you that we were expecting that maybe Nardinella might be closely related. Nardinella would be housed in closely related molotines, weevils. And what we ended up finding was that was mostly the case. So all of the Nardinella that I found was hosted in molotine weevils, and they aligned in the tree with Nardinella that was also hosted by molotine or cryptorhamne kinase. Sorry. As compared to Nardinella that was hosted by more distantly related weevils like entomine weevils or Bryopterine weevils. Another fun finding was that I found that Gastrotachrus and Starius, their strains of Nardinella, were more closely related to Nardinella hosted by other molotines or other cryptorhamne kinds than to Nardinella that was hosted by Stromius. And just to remind you of what that looks like in the evolutionary context of weevils. So the Nardinella that I found that was hosted in the GLSS weevils was closely associated with Nardinella hosted by these guys, so the purple arrows, and not closely associated with Nardinella hosted by these guys, entomine with kind of green arrow or Bryopterine with red arrow. What I found with Sodalis was it was pretty consistent with it being a promiscuous microbe. So it was a bit all across the board. We were finding closely, there was like a cluster of Sodalis here and a different cluster of Sodalis here. And so there was distantly related weevils hosted closely related Sodalis in this cluster. And then there was some closely related weevils in cluster A and cluster B. So the relatedness didn't seem to have anything to, the relatedness of the host weevil didn't seem to have anything to do with the relatedness of the microbe. And then I wanted to answer a couple other questions. So I wanted to know, did habitat impact symbiont occurrence? No. So both Nardinella and Sodalis could be found in either habitat. And then also I had initially hypothesized that there would be a more conserved microbiome in weevils that were Sericinoides, weren't restricted to the shoreline niche, and that's not what I found. There was no significant difference in richness in weevils in any of the different habitats I sampled. And so what this might kind of look like, if we were to look at it graphically, is that the amount of diversity of the microbes in either habitat was not that different. And then, did storage condition impact symbiont detection? The answer was no, it didn't impact symbiont detection. I was still able to detect symbionts, whether or not it was collected from a live caught weevil or from a pin museum specimen. This is just a principal component analysis where in purple we have the museum specimens and in orange we have some of the live caught specimens, and there's a great amount of overlap. Conclusions. Here we are, we're at the end. So what I found with the phylogeny was that Stremnius is not sister to Asterias, Gastrocapris, and Lobosoma. This is the tree that I thought I was going to find. And this is the tree that I actually found. I found Asterias and Gastrocapris really closely associated. Lobosoma is sister to that group, and Stremnius was not closely associated or not sister with that group. Okay. I also found that genus Gastrocapris should probably be synonymized with Asterias. And also that it's hard to say what the sister to the Gastrocapris Lobosoma could be. My biggest takeaway here was that even small, seemingly monophyletic clades within weevils require investigation with molecular characters in order to better understand the weevil tree of life. With the microbial data, I found a lot. I learned a lot. So I found that P. weevil endosymbionts can be obtained from adult whole body weevils that were housed in museums. I also found that habitat was not the underlying factor driving symbiosis. And I also discovered an endosymbiont, maybe near Curculionophilus, found in one of my specimens. So far, this appears to be the first report of a Curculionophilus ally outside of the Curculion group, which is really cool. My biggest takeaway, though, from the microbial part was that the microbial community structure can provide an extra layer of information necessary to understand the weevil tree of life. Future directions. In the future, I would love to investigate some further morphological characters of the weevils and increase my taxon sample size. In particular, I would like to look at the general morphology of Asterias and Lobosoma in comparison to Stremnius and Plinthus, because those are likely diagnostic characters. They are diagnostic within, or they are shared and derived within different Plinthus groups. So maybe that we could compare Plinthus and Stremnius and see if that helps us to understand if there is a relationship there, and then what does that look like in comparison to the others. I'd also like to test the monophyly, including some other things that have been allied with the Gastrocapris, Lobosoma, and Asterias based on morphological characters in the past that haven't been looked at in terms of shared derived morphological characters, and that I didn't include here in this molecular phylogeny. So those are things like Protoclinus, Protoclodes, Doramazo, and Protocales. For microbial stuff, I would love to get more Stremnius samples. So I only had one Stremnius stremani that I found this really cool, interesting endosymbiont. So I'd like to increase my sample size there. And then it would be really cool to do some fluorescent in situ hybridization imaging to see whether or not there is a bacterium in Stremnius stremani. And where is the bacterium? Because it might help us to identify what endosymbiont that is, because if it was in the more anterior bacterium, maybe that is evidence that it would be Sodelis. And if it's in the more posterior bacterium, maybe that's evidence that it would be Curculionophilus. And then, now that I know that you can use museum specimens, use Sequenas amplicon sequencing, which is a little bit cheaper than whole genome sequencing and other methods that you could use to get sequence data out of dried museum specimens. I would love to just canvas as many molotine weevils as I can in museum collections for what endosymbiont they have. And it's a really cool idea because with this, you would be able to look at things in a larger evolutionary context, because museums have already a huge store of molecular data there that's already been identified, it's been collected from all around the world. So you could just sit down in the museum and essentially look at the weevil tree of life in front of you and start sampling from there. And I think that would be a really fun and interesting project. No. No, I want to give a really effusive thank you. I was told that I am allowed to spend at least 5 minutes giving thank yous, and I will take my time saying so many thank yous. I would like to first say thank you to my committee members who have been really, like, I've tried to give this talk a bunch of times, and every time I have literally not been able to find words enough to thank my committee. And so I'm going to try here, but just know that my committee members have helped me so much. I had to take an extra year to complete this master's degree, and I would not have been able to finish without my committee members. So I want to say a huge thank you to Becky and David and Lupe and Bob. I could not say thank you enough. I also want to thank the staff members here at the Department of Integrative Biology, Melissa, Tracy, Teresa, and Tara, who have answered a kabillion questions, like a kabillion. The Madison Lab, other members of the Madison Lab, here we all are. So there's Cassie and Arden and Olivia and Camden and Connor and Lucy and some past members of the Madison Lab, well, Rhea and Cassie as well, who all helped for encouragement, helped me do sequencing, helped me with creating some of these matrices that I made. Here's some more pictures of the lab. I also wanted to thank the Vega Thurber Lab because I would not have done any of the microbial stuff without the Vega Thurber Lab. All of the 16S sequencing, or a good portion of the 16S sequencing, was done with that lab, and also preparing for PCR for the sequencing was actually done by Alex Mompe. So like, they were just so helpful, so kind. They shared resources, they shared anecdotes, they answered a million questions. They took me on a lab retreat. Thank you. I also wanted to thank the Bi22X teaching team. I found that anytime that I was discouraged with working on this project, I could find a lot of solace in teaching, and I learned throughout this 3 years how much I love teaching. To be honest, I did not think I liked it to begin with, and by the end of my time here, I was like near tears having to leave teaching the Bi22X series, and I just really appreciated the entire teaching team for being there, especially Lori and Carmen. Here is just us doing some field expeditions with the Bi22X group. It's not a great photo because you can't see any of the students' faces, because I thought that that would probably be a breach of confidence or something, but it was a good time. I also wanted to thank my partner, Devin, for listening to this talk a million times, reading my thesis a million times, and being super supportive. My family, this is my dad, my mom, and my brother. They actually came out to Oregon to visit me, and even though they do not like bugs at all, they do really love me, and so I really appreciate them. I have been able to go across the border back to Canada to see them. Yay. I also wanted to thank friends and more friends. Because I would have gone insane a million times without them. And finally, I wanted to thank my cat, Goo. Okay, you can now applaud.
We have a few minutes for questions from anybody but the committee.
Yay, okay, what?
So on the maximum line picture that you created for the endosylums, you mentioned that you're surprised the PENC group had such a long branch. Maybe kind of a simpleton question, but like, what does a really long branch picture like this suggest biologically?
Yes, okay. So the branches here in phylogenetic trees are measuring basically the difference between sequences, how many bases are different from one another. And so what we're seeing here is that Sodalis, all of these have really short branch lengths together, so the sequences are relatively similar to one another. This really long branch is showing that, okay, yeah, they are maybe similar to Sodalis, but the sequences are really different from the Sodalis sequence. Oh, sorry about that. Yeah. Does that answer your question?
Yeah, so their sequence is much more divergent. Okay,
The molecular characters that are available seem to be quite different. Yes.
The question on Zoom, it says, with museum specimens, how accessible are the beetles to take for DNA extraction? Does the beetle get destroyed in the process?
Yeah, okay. That's one of the things I'm really excited about. So I didn't want to destroy the weevils when I was doing this project. And so I was using whole body weevils from the museum so that I wasn't like crushing them up using different parts of them. And the concern with that is that it limits my ability to say that something is certainly an endosymbiont, right? Because I didn't get it from the internal part of the organism. Maybe that was a microbe that was just on top of the weevil. Maybe it was a microbe that was just kind of nearby the weevil at the time. But what I found here was that I could use the whole body insect. I could still find a known endosymbiont that have not been found free living in super high relative abundance, which would imply to me, or is pretty suggestive that it is an endosymbiont. And I wouldn't have to destroy the museum specimen. And it depends on the institution. Some institutions are okay with you destroying their material, especially if it's something that they have a lot of. But if I wanted to canvass like all of the tribes of Malatini, there are some things that are going to be a lot more rare. I'm not going to be able to find a lot of specimens of them in museums. And where I am able to find them, they're probably going to be like, please don't destroy this single specimen I have that's representative of that group. Yes.
Oh, actually, so you said that you personally sequenced a lot in all the blue in this tree. And from insider knowledge, I know it was a lot more than just that blue. How much of the weevil phylogenetic tree did you personally sequence for this project?
Well, for the weevil phylogeny, I would say that the tree that we see here, it was about 50-50. So half of, let me go over to that tree. So half of this tree were data that I collected from online sources, other papers, and half of it was sequences that work that I did myself. I will say that there was a lot more weevils than are on the tree that I did sequence myself that just didn't make it into the final cut, or me and the other lab members did all the work to like extract the DNA and go and do the PCR and send off the thing for sequencing, and it didn't work. I found there was at least 2 genes that we tried multiple times to get sequence data from, and it was not working. And so there was a lot of sequencing that was wasted. Yeah, that is, yes.
Congrats, Nat. This is such a cool talk. I was wondering why you think that Asterias tenoides was so much harder to find than you initially thought.
Okay, I have like a lot of conjectured thought about this, but my main thought is just that a lot of the occurrence records for Asterias tenoides in Oregon and Washington and even Northern California, Asterias quadrilaterus were found there. They're older occurrence records. And so I think that in the last 100 years, there's been a lot of development of the coastal region of the Pacific Northwest. And so there's not a lot of wood anymore on the coast. And so if you go and look at these beaches where I was expecting there to be like lots and lots of driftwood and lots of diversity, there's not a lot of driftwood. And there's also a lot of heavy foot traffic. It's destroyed areas or disturbed areas. So I don't know. But if I was to guess, I would say that I think it's just the changing landscape. And also from collaborators, I know that it's really easy to find Asterias tenoides in Alaska and Russia. And I'm thinking that maybe the landscape there, the coastal landscape, is still, very vibrant, lots of wood, undisturbed areas. Yeah, I don't know. Yeah.
I guess in terms of going off of that, like, did you try using Bug Guide or iNaturalist to look for observation? And was there anything that you were like really hoping to find that you just never did? Like, I know you found tenoides once, but was there anything you're like, I wish I could have found that?
Yeah. Really good question. I did use iNaturalist and Bug Guide. I love iNaturalist and Bug Guide. A lot of what I was doing was exploratory. So going into it, there wasn't like, oh, I hope to find a whole bunch of this weevil outside of my core group. So otherwise, yeah, there was a lot of like, I'm not finding Gixotappers and Asterias and Lopazoa. There's a lot of that. But outside of that, it was an exciting discovery every time that I was finding other driftwood weevils that were out there to compare with. Yeah. That answers
Yeah,
your question.
no, that's,
Yeah.
that's, I think it's really awesome that you were able to like you collected a bunch of different weevils from the driftwood. I think that was really great to like balance against like the core group that you were looking at for sure. And just like characterize that really interesting niche community. Like I would never think about weevils living in driftwood on the coast. So that was really interesting.
If I were to do this study again, I would like to do more nighttime sampling. I'd never sampled at night, and likely these creatures are more active at night. So that might also be another reason, A, that I wasn't finding a lot of them, and B, an area that I wish I could have gone to, like time of day that I wish I would have gone to, and maybe it would have found more.
Last question. Did you find, curious about the genes, did you find one that sort of drove the analysis more than others?
Ooh, okay, so this is kind of a cop out to that, but I would say that I got a lot of information out of CO1. And the reason for that is just because it's a known barcode in insects in particular. And so there's just a lot of data to be able to compare to. So it just in terms of taxon sampling, it worked out the best. It was also relatively easy for me to sequence. So I also was able to contribute more data to that. I would say conversely that I was limited by EF1 alpha because there is an intron in that gene, which made it really painful to do alignment for a long time of manual alignment. It also made it so that things online there was not a lot to compare with online because either they had done their alignment differently, they included the intron, they included the exon, differences like that, or they just no one else was also having success with sequencing that gene, and so there was not a lot of comparable information. CO1 best, EF1 alpha first.
Okay, well, with that, let's thank Nat one more time. So now we're gonna, now we're gonna transition to, well, a break, short break, but then committee will go to room 2029 and we'll have the rest of